Hello, Brain Shakers. Welcome to the Brain Shakers Academy. Brave Alistair's here. Now, in today's session, we're going to be looking at yet another exciting and interesting topic under gametogenesis, which is the process of a female gamete formation. Now, we have looked at male gamete formation, or how the spermatozoa is formed, but in today's session, we'll be looking at how the female ovum or the egg is formed, how it matures, and then how it gets to be released through that process of ovulation. Without wasting much time, let's get right into it. Like in spermatogenesis, the process of female gamete formation or oogenesis also begins around the fourth week of gestation, where the PGCs or the primordial germ cells migrate from the wall of the yolk sac into the ovaries, where they then differentiate into oogonia. Now, the oogonia will then later undergo mitotic divisions, where the parent cell or a parent oogonia is going to separate and divide into two daughter cells, and this then populates the ovary. The oogonia that is then in the ovary will enter the first meiotic division and undergo DNA replication to form the primary oocytes. Now, by the fifth month of fetal life, the primary oocytes are well formed, and hence there is no further oogonia that is there, and that makes the baby that is born uh, having or containing no oogonia, but rather primary oocytes. Because the primary oocytes have entered meiosis 1, they enter into the prophase, and because we looked at the meiotic divisions as having a stage, or the first meiotic division having a longer prophase that has five subphases, where we have the leptoten, the zygoten, the pachyten, the diploten, and the diakinesis. The primary oocyte that has entered into the first meiotic phase is going to enter into the prophase. It will go into the leptoten, the zygoten, the pachyten, and when it reaches the stage of diploten, it undergoes its first arrest. So these primary oocytes are now going to be arrested in the diploten stage of meiosis 1 and will stay in there until puberty. Now, after puberty, this is where now the primary oocytes will begin to mature with each and every ovarian cycle, and usually only one reaching a full maturity. Now, when the cells or the primary oocytes are undergoing the development into a mature oocyte, they undergo a number of stages. Now, let's get into those stages that the primary oocyte will have to undergo for it to be mature before it can then be released from the ovum and during the process of ovulation. During every ovarian cycle, you notice that the primary oocyte will have to complete its first meiotic division to form two daughter cells, where there will be formation of the secondary oocyte and the polar body. The polar body will obviously degenerate, but the secondary oocyte is what may then go into the second meiotic division. Now, the primary oocyte, for it to develop into what is referred to as a mature oocyte, will have to undergo three phases, which is the pre-antral stage, the antral stage, and the pre-ovulatory stage. Now, in the pre-antral stage, what is going to happen basically is that the cell is going to grow exponentially in size, and then you have follicular cells that will grow and proliferate to form a stratified cuboidal epithelium. Now, if you look at this particular cell here, you'll find that there's this yellow part here, and then you have, just after the yellow part, you have a green line there, which is a zona pellucida. Now, around the zona pellucida, you have what we are referring to as the granulosa cells. Now, the granulosa cells is just a group of cuboidal cells that are put in layers, and that is why it is referred to as a stratified cuboidal epithelium. To stratify is just to put in layers. So the granulosa cells now are responsible for secreting the glycoproteins. Now, the glycoproteins are a chemical form that is responsible for the formation of the zona pellucida, which is just this green layer that we can see right in the diagram here. That is the zona pellucida around the primary oocyte. Now, after you have noted that the zona pellucida has formed and you have granulosa cells, around the granulosa cells, on top of the granulosa cells, you have a connective tissue of cells or connective tissue cells that also then differentiate to form what is referred to as the thicker folliculi. Now, the thicker folliculi is a specialized layer of surrounding cells that is very responsive and very sensitive to luteinizing hormone. Now, just like the process of spermatogenesis, the process of gametogenesis in females is also regulated by the HPG axis, which is the hypothalamus pituitary gonadal axis, where the hypothalamus is inputted to produce the gonadotropin releasing hormone, which then affects the anterior pituitary gland to produce its hormones, which is the luteinizing hormone and the follicle stimulating hormone. Of importance now is the luteinizing hormone coming to play a bigger role on the thicker follicular cells, which is just a connective tissue cells that forms on top of the granulosa cells. Now, what does the luteinizing hormone do once it comes into contact with the thicker follicular cells? It means that it comes to the thicker follicular cells and stimulates them to produce androgens. Now, androgens are important in eliciting sexual pleasure, in eliciting an increase in the libido or sexual appetite. And this is why when there is a surge prior to ovulation, when there is an increase in the luteinizing hormone, a female prior to ovulation will have an increase in the sexual desire, so that there could be a spermatozoa that will then fertilize the ovum once ovulation has happened. So that all begins in the pre-antral stage. Now we go to the antral stage, which is the second stage. Within the granulosa cells, you will notice that there will be formation of the fluid-filled spaces here. Now these fluid-filled spaces, they will begin to coalesce and form a central fluid-filled space, which will now be referred to as the antrum, and hence the stage being called the antral stage. And the only remaining thing now is going to be more or less like a connecting stalk between the cells or the granulosa cells that surround the zona pellucida and the other granulosa cells that surround the rest of that oocyte. And this connecting stalk is what we call the cumulus wolferus. Now, once you have this particular secondary follicle now, because the follicle now has an antrum and it is referred to as a secondary follicle, and in each monthly cycle, one of these secondary follicles is going to become dominant and then later developed further under the influence of the follicle stimulating hormone and the luteinizing hormone, as well as the estrogen that is going to be produced. Then you get to the pre-ovulatory stage. Now, in the pre-ovulatory stage, we've already talked about the surge of the luteinizing hormone, or the increase in the luteinizing hormone, meaning that now what is going to happen is that that oocyte is going to complete meiosis 1, and as it completes meiosis 1, then there will be production of one secondary oocyte and a first polar body. The first polar body will then also undergo a series of cell division, which is the second meiotic division, to give you two polar bodies. And then you have the secondary oocyte that will also undergo meiosis 2 and giving rise to another polar body. But then the secondary oocyte has the second arrest of the cycle in the metaphase, that is in the second meiotic division, and this usually happens 3 hours prior to ovulation, so that the body now prepares itself, prepares this particular egg for fertilization. So the arrest is going to happen in the metaphase, and then now you get into the stage of ovulation. When you get to the stage of ovulation, it has to be the luteinizing hormone. The surge in the luteinizing hormone now is what is going to increase the collagenase activity. Now, collagenase is just an enzyme that has the capacity to degrade the collagen around the ovum, so that it makes the wall weaker and creates a weaker point, so that when the ovarian wall muscular contractions begin, then it can push that ovum through the weaker point out of the ovary, and that is how the egg then is going to be released. So all this is controlled by the luteinizing hormone. Now, remember, we also mentioned that the luteinizing hormone is also increasing the production of the androgens that increase the sexual desire, so that once then the ovum has been released, then this person who has a higher sexual desire at this particular time could have met um, could have met a man, and then they end up getting into coitus, and then a spermatozoa is deposited into the vagina for the fertilization process to happen. Now, what happens is that if then the ovum that has been released out of the ovary has then been picked by the fimbriated end of the fallopian tube, then it is going to be waffled straight into the fallopian tube and mostly into the ampulla region, which is the largest portion of the fallopian tubes, awaiting fertilization to take effect. Now, if fertilization does occur, those peristaltic movements then will push the fertilized ovum straight into the uterus for nidation or implantation. But if fertilization does not occur, then the oocyte is going to degenerate in the next 24 hours after ovulation, and it will remain arrested in meiosis 1 without completing meiosis 1, or without even getting into the second meiotic division, and then later on be eliminated through a process of menstruation. So the process of fertilization is what completes the process of female gametogenesis, or rather oogenesis, because it is only after fertilization that now that mature oocyte will get into the second meiotic division and then complete the process. So that is basically what happens around the formation, the maturation, and then the release of that ovum in readiness for the process of fertilization to take effect. Now, if you found this particular video as helpful in understanding the formation, the maturation, and then the release of the ovum, please give it a thumbs up and share this video as much as possible. And don't forget to subscribe to our YouTube channel, which is Brain Shakers Academy. And as always, it's a pleasure to have you here. Thank you so much for watching, and I will see you in the next one.